Okay, welcome back. This week we're going to start talking about cellular respiration. So we just finished up our unit on photosynthesis, and if you'll remember correctly, that was a series of redox reactions that ended in the production of G3P. G3P being a reactant that the plants then use to produce glucose. And within glucose, within the carbon bonds, are lots and lots of energy. And so that's stored in our autotrophs. And so our autotrophs can use that glucose when it needs energy. And then the heterotrophs, when they ingest the autotrophs, also consume those glucose products as well. And then how we get the energy out of that involves cellular respiration. So why do we eat and breathe? Obviously, we do lots of things that expend energy. So anything from just moving your finger to the more exciting things, your hobbies, mine for instance, whitewater kayaking. All of that requires energy. And so we need to ingest molecules that hold a lot of energy, such as glucose that we talked about in photosynthesis. And then the short answer of why we breathe is because we need oxygen to be a final electron acceptor in cellular respiration, and we will talk in detail about that. But why we eat and breathe basically boils down to cellular respiration. So let's just go ahead and be clear that respiration is not breathing. That is one mistake that people often make. What we mean at the very basic level is that respiration is the production of ATP from organic molecules, in other words, from food. And so, of course, where we get that is the breathing part is important, because as we breathe, we take in oxygen, which we've already said was an electron acceptor, and so that is key in cellular respiration. And then we get our glucose from the breakdown of food that we eat. So oxygen and glucose are reactants for what we call cellular respiration. So the mitochondria is the organelle that is the site of cellular respiration. Whether it be in plant cells or animal cells, cellular respiration takes place here. And each mitochondria is bound by two highly specialized membranes. And because of those, they create two separate spaces, the intermembrane space and the matrix. And each one of those plays a very significant and different role within cellular respiration. So the outer membrane has what we call porins in it. That's a particular transport protein, and it's pretty large, and it allows small molecules to go through pretty easily. So it kind of resembles like a sieve, and permeable to all molecules of a certain size. So because of that, the intermembrane space doesn't look a whole lot different than the cytosol in terms of a lot of those small proteins. However, that intermembrane is highly impermeable to quite a few things. And so that makes the matrix very different from the intermembrane space. One thing that's important about that inner membrane is that it's impermeable to ions, and we'll talk about why that's important later. But just so that you understand that the chemical makeup of the two different membranes serve their purposes in the role of cellular respiration for these two areas that are so important, that matrix and the intermembrane space. In its simplest form, cellular respiration is the breaking down of glucose, which is an exergonic reaction, and that is coupled then with ATP production, which is an endergonic reaction. So that goes from its reactants of ADP and phosphate into ATP. So that is the coupling of an exergonic and an endergonic reaction. Obviously, it's not just two reactions, it's multiple steps in that, and we're going to talk about that. But then those ATP molecules have all that energy stored within those phosphate bonds that we talked about, and that is used then to power various cellular processes. So taking potential energy in the chemical bonds and transferring that to either processes such as transport or mechanical processes, those would be kinetic energy, or converting it to another form of chemical energy within the cell. So that's our overview of our cellular respiration, the very basics of it. So let's just very briefly look at the relationship between photosynthesis and cellular respiration. They're basically, if you look at it in its simplest form, it is a specific reaction in one direction for photosynthesis, and then the breakdown of it in cellular respiration. So if you'll remember correctly, with our photosynthesis, we drove this equation going from carbon dioxide plus water equals a sugar, oxygen, and water. And that's driven by light energy, and it takes place in the chloroplast. When we look at cellular respiration, we're taking the products of photosynthesis, so glucose and oxygen and water, glucose and oxygen, and then we're breaking it down to carbon dioxide and water and a lot of ATP. So about 38 ATPs. So this is a great relationship between photoautotrophs and heterotrophs. Having said that, cellular respiration does also take place in plants, so let's not forget that. So we have our photoautotrophs that take in the carbon dioxide, they put it into a useful form that stores a lot of energy in the form of glucose, and they release oxygen. Well, that's super convenient because those of us who ingest them also require oxygen to breathe, and so we then will take in the oxygen and the glucose, and the byproduct of cellular respiration then is carbon dioxide and water, which can then be recycled into photosynthesis again. So there's this cyclical relationship between these two processes. So here is our very simplified version of cellular respiration, and I'm sure as you learned from our photosynthesis lectures, we know that this is just a summary of all the different reactions that happen, and we're going to go into detail with all the different reactions. So for cellular respiration, we begin with glucose and oxygen, and through a series of reactions we get carbon dioxide, water, and ATP. And again, this is a redox reaction, so our glucose becomes oxidized into carbon dioxide. Remember, we said oxidation is loss, so we have lost electrons and lost hydrogens. On the other hand, the oxygen that is supplied gets reduced to water. So reduction is gain, so it gains electrons and hydrogens. So this is our oxidation reduction reaction. And like I said, this is a summary of cellular respiration. So some molecules within the cell are high energy intermediates, or there are electron shuttles or electron carriers. So I like to use this analogy where we have energy from the outlet, and that outlet donates electrons to a battery. And this battery, once it is filled with electrons, is full. And then the battery itself can then donate electrons to an electron acceptor, in this case our lights, our battery operated lights. And as those electrons are pulled from the full battery, then we have an empty battery. And there are molecules that act similar to this within a cell, and certainly within our cellular respiration process. So like our battery analogy, two particular molecules that we're going to be using over and over again are actually what we call coenzymes. So we have NAD and FAD. And these two get cycled over and over again. So let's just take NAD plus for instance. It's empty, meaning that it does not have much energy. So it goes through a reduction process, and in reduction process it gains electrons and hydrogen ions to become NADH. At that point it is full, it is an electron carrier, and it can then donate its electrons to any electron acceptor. And when it does that, that process is called oxidation, and it goes back to the form NAD+. That's the one that's used most often. FAD is also used as a coenzyme, and again it goes through reduction in which it gains electrons and hydrogen ions and becomes FADH2. That is like the full battery form, and it can then donate its electrons through the process of oxidation to another molecule. So these are the two coenzymes. Well, there are multiple enzymes and coenzymes throughout our process, but these two are important. They're used over and over again in cellular respiration. So here's just another look at those two coenzymes. I wanted you to be able to at least picture what they look like. Our NAD plus on the left and FAD on the right in blue. Crucial coenzymes for cellular respiration. So I'd like you to take a minute just to click on over to this link to look at how the coenzyme NAD plus works. I wanted to embed it in the video, but for some reason it will not allow me to do that. So I will also include it under extras on your Sakai site as well, but you can copy and paste this into your browser and just take a look at that. So let's get down to the meat of it. Let's talk about cellular respiration. There are 4 phases to our cellular respiration. So the first one is glycolysis. And if you look at our diagram over here on the right, you'll notice where all these phases are taking place. And you'll note that glycolysis is on the outside of the mitochondria. So even though the majority of these reactions are going to occur in the mitochondria, that first phase, which is kind of like the setup phase, actually happens in the cytoplasm. So glycolysis is an anaerobic reaction, meaning that it does not require oxygen, and it takes place in the cytoplasm. And essentially, glycolysis is just the breakdown of glucose into 2 molecules of pyruvate. And then those pyruvate molecules get moved over into the mitochondria. And so our next 3 phases are going to take place in the mitochondria, and they're going to be aerobic reactions. Aerobic reaction just means that they require oxygen. So the second step is what we call the preparatory reaction, or pyruvate oxidation. The third is the citric acid cycle, or what we call Krebs cycle. And then the fourth is the electron transport chain that we've talked about in relation to photosynthesis as well. That's where all of our ATP is going to be produced. So let's get started with these reactions. So here are the steps that glycolysis follows in order to produce 2 molecules of pyruvate. And the way we have this broken down here is into energy investment steps and energy harvesting steps. And that just has to do with does it require ATP or produce ATP. And our first section up here, what we do at the very beginning is we phosphorylate glucose. And so in order to do that, we require ATP. So that's an energy investment step. And at the end of that, we are going to produce 2 molecules of G3P. And that should sound familiar because those were the products that we came up with after photosynthesis. So we're going to look a little closer at this energy investment step, but just wanted you to see this overall view. So glycolysis is actually a series of 10 steps, so it's a metabolic pathway, and each one of these steps has its own enzymes that allow it to occur. So if you think about all along the way, this pathway could be sped up based on what you know about enzymes by increasing the temperature a little bit, by increasing the substrate concentration, or it could be inhibited in any of these steps by an inhibitor coming in on the active site on any of these enzymes, or if we had a non-competitive inhibitor, which would bind to the enzyme in a different site, but then that causes the changes in the active site, if you'll remember that. So be thinking about that as we look at glycolysis and just how intricate it is with these 10 steps, with all these enzymes and the coenzymes that go with it. And I think you start to get an appreciation for just sometimes how miraculous it is that all this takes place within every cell of your body, which is so many. So let's look right now at, okay, so let's look a little closer at the energy investment step of glycolysis. So from the very beginning, we've eaten our food, our digestive system has done its part, broken it down and converted it to glucose molecules. Glucose is just a 6-carbon molecule. We've talked about that before. And so glucose has transporters in the plasma membrane that allow it to come in pretty easily into the cytosol. So that's where we pick up our story here. So on the very left, you can see our 6-carbon glucose molecule right here. And as soon as it comes in, it's going to be what we call phosphorylated. So we're adding a phosphate group. And this actually takes 3 different enzymes to accomplish that goal. And so anytime there's an enzyme that is adding a phosphate group to a molecule, it's called a hexokinase. So a couple of hexokinases are active in these steps, as well as isomerases. Can you think of what that means based on what you know about isomers and then the ending aces? Essentially, that means an enzyme that is converting a molecule into a different shape, the different isomer. So I just wanted to make sure that you knew this wasn't just one step. This is a series of 3 steps that require 3 separate enzymes. And while it's doing that, it is using up 2 ATP molecules in order to add these phosphate groups to the glucose molecule. And what that does for us is once the glucose molecule is phosphorylated on both ends, it is then kind of trapped in the cytosol or in the cytoplasm of the cell, so it won't be leaking back out. Now, one thing to note is if our phosphorylated glucose, or our G6P, so glucose is a 6-carbon molecule with phosphate on it, so if our levels of G6P increase, they can hit a threshold level that will then go back and make sure that these enzymes are turned off. In other words, if we have too much G6P, the process of glycolysis is going to stop because it's signaling the rest of the body, I have enough, I don't need any more right now. Okay, so that's a feedback loop. And that's how regulation happens so that we can maintain homeostasis. Okay, but how do we get this ball moving forward? Well, then we're going to go through a next step with another enzyme, and this enzyme is called aldolase. And aldolase is essentially going to cleave that glucose 6-phosphate into 2 3-carbon phosphorylated molecules. And there is a little nuance here, but you in the end wind up with a G3P. And again, that should sound familiar to you because G3P is something that we ended up with at the end of photosynthesis. So this is our energy investment step. So we have used 2 ATP molecules so far, and we've come up with I can't speak. Okay, 2 molecules of ATP, and we are yielding 2 molecules of glyceraldehyde 3-phosphate. Okay, so our next series of reactions in glycolysis is what we call our energy harvesting steps. And that just means at the end of these series of reactions, we're going to have a net gain of ATP being synthesized. So we're going to start with G3P, And remember, we have two of those because at the end of our energy investment step, we had lysed glucose through a series of steps into G3P, which is our glyceraldehyde 3-phosphate. So where we pick up on that is that we are going to begin with the oxidation of G3P. Remember, oxidation is loss, so we are removing electrons and the accompanying hydrogen ions. And then in a coupled reaction, we are taking NAD and reducing it to NADH. So if you'll remember when we looked at the battery analogy, the NADH is the molecule that is like the full battery. And in fact, we're going to see later that the NADH is going to carry 2 high energy electrons to the electron transport chain. And when it does, it's going to then give up its hydrogen ions and its electrons, and NAD+ will be recycled. So this is just the beginning of that. In this same step, we have an inorganic phosphate group that is going to be added to our molecule. And so that is going to give us 1,3-bisphosphoglycerate, which we also call BPG. Okay, so that's this right here. Keeping in mind, each one of these steps takes at least one enzyme. Okay, so what we're going to do now is, because of this inorganic phosphate group, it is high in energy, and what we're going to do here is we're going to do what we call substrate level ATP synthesis, or substrate level phosphorylation. So in other words, the ADP that is sitting around, we're going to donate this inorganic phosphate group to it and produce 1 ATP molecule. And when we do that, now we've also produced 3-phosphoglycerate, or 3PG, which is another 3-carbon molecule. And so we've done this for each of these sets of reactions. So right at this point, we have created 2 ATP molecules. I am terrible with writing with this pen, so I apologize for that. So in the previous slide, we talked about substrate level phosphorylation, and I just wanted to talk a little bit about that so that you understand how that differs from the other types of production of ATP. So anytime we say we are phosphorylating something, it means that we are adding a phosphate group. And often we think about the production of ATP. And that is not the only phosphorylation that occurs in the cell, but for our purposes, that's what we're going to concentrate on. So there are 2 different types of phosphorylation that we're going to focus on. One is substrate level phosphorylation, and the other is oxidative phosphorylation. You're actually already familiar with oxidative phosphorylation, whether you knew it or not. But I went ahead and actually wrote down the difference between the two, just because if you want to come back later and go, now what exactly did she say? I just wrote it completely out for you. So during glycolysis, outside of the mitochondria, in the cytoplasm, we can accomplish substrate level phosphorylation. And all that means is that there is a physical transfer directly from a phosphorylated molecule to ADP in order to produce ATP. And it does involve an enzyme, just like you see in this image here. Now, the other type of phosphorylation is oxidative phosphorylation, which we've already seen in our chloroplast, and we're going to see in our mitochondria as we move forward into the other parts of cellular respiration. And essentially what that is where we have a oxidation reduction reaction producing ATP. So in other words, the nutrients or the chemicals provide the energy to transfer a phosphate group to ADP. And the method for doing that is through the electron transport chain that we've been talking about. We saw it in our photosynthesis reaction. We're going to see it again in our cellular respiration. So I just wanted you to know the difference, because as we've just done glycolysis, we've produced ATP through substrate level phosphorylation, but in the rest of cellular respiration, we're going to switch gears and use oxidative phosphorylation to make ATP. So this is a summary slide for glycolysis. So we started out, we put in glucose molecule, 2 ATPs, and we have put in 2 NAD plus molecules. And out of it, we've gotten 2 pyruvate molecules, 4 ATP molecules, and 2 NADH molecules. Now, just to clarify, we have produced 4 ATP, but since we put in 2 ATP, that gives us a net production of 2 ATP molecules. And the NADH molecules are going to be used further down in the electron transport chain, and we're going to take those pyruvate molecules and continue our process of cellular respiration. So this video is a summary of how glycolysis works. You can either cut and paste this URL into your browser, or I've also included it under the extras on your Sakai site. So check that out. So now we're moving into the second phase of cellular respiration, and that's the preparatory reaction. And essentially what that means is we are preparing the products of glycolysis to move into the citric acid cycle. And so if you'll remember with our glycolysis, for every glucose molecule that we broke down, we wound up with 2 pyruvate molecules. And that took place in the cytoplasm. So now we're moving into the mitochondria. And if you'll look at it with me for just a moment, we said before that we have 2 membranes. The inner membrane has lots of infolding, and we call that the cristae, and then the very inside, which is the matrix. And the matrix, right here, is the location of this preparatory reaction. So how do we get pyruvate into the matrix? Well, it's this lovely molecule called pyruvate translocase. And just as the name would suggest, that is an enzyme that moves pyruvate from the cytoplasm into the inside of the mitochondria. I love that name just because it's so simple. Pyruvate translocase. It's exactly what it does. Okay, so just to give you a sense here of where we're going, that preparation and the citric acid cycle are both going to take place in this matrix. And then the products of that are going to then be used for the electron transport chain, which is all along these cristae. And so if you remember, all those infoldings increases the surface area, so we have lots of places to create ATP. And in fact, since this is where so many ATPs are produced, oftentimes the mitochondria is called the powerhouse of the cell, because that is where all the power is made. So what's going on with this preparatory reaction? First of all, like we talked about, we're going to transport pyruvate into the mitochondria using our pyruvate translocase enzyme. And then we're going to start by basically oxidizing pyruvate, which means, remember, oxidation is loss, so we are getting rid of our electrons and our protons, and we're donating those to NAD+. So NAD+ is reduced to NADH plus hydrogen ions. And then we're also going to get rid of a carbon. So we're taking a 3-carbon pyruvate and we're changing that to a 2-carbon acetyl group. And when we do this, we lose one of those carbons in the form of carbon dioxide. In addition, we're going to then add what we call a coenzyme A, and we're going to add the acetyl group to that, so that at the end we have what's called acetyl-CoA, and that's going to move into the citric acid cycle. So this process occurs for every pyruvate. So in glycolysis, when we broke down 1 molecule of glucose, we wound up with 2 pyruvate molecules. So for every glucose we break down, we wind up with 2 carbon dioxides, 2 NADHs, and 2 acetyl-CoAs. So where do all the products go from the preparatory cycle? First of all, our carbon dioxide, remember with 1 molecule of glucose we have 2 carbon dioxide produced, and this diffuses back out of the mitochondria, back out of the cell, into the bloodstream, and where it is carried to the lungs, where we can breathe it back out. So it's sort of a waste product. The NADH, remember that's like our full battery. It has lots of high electrons and high energy electrons and protons. That's going to move to the cristae of the mitochondria, where it can then be used in the electron transport chain. The acetyl-CoA is going to be part of our Krebs cycle or our citric acid cycle, and so that's going to remain in the matrix of the mitochondria. So that's where those are going. And then we're going to stop here for now so you can review all this, and then we will pick up with our next lecture and go on into the citric acid cycle.